Good morning, students. Welcome back to another session of Plant Physiology, Chapter 4, Photosynthesis in Higher Plants. In today's class, you are going to learn about chemiosmotic hypothesis. This is very important topic. If you understand, then you'll have clear idea about the light reaction. The most important thing about light reaction is it takes place in granum thylakoid, in the thylakoid membrane, which is located in chloroplast, and it will be utilizing the sunlight energy, water, and synthesizing ATP, NADPH, as well as oxygen is liberated. So these are the things taking place at the granum thylakoid. So far, in the previous classes, you have learned how the cyclic photophosphorylation, non-cyclic photophosphorylation, that means the synthesis of ATP is taking place. You have learned. And you know about the photosystem 1 located at P700 nanometer wavelength, and photosystem 2, that is P680, that is at 680 nanometer wavelength, how the reaction center is located. You have learned. And in today's class, you are going to understand in a clear way how ATP and NADPH are synthesized, what actually is happening. You know only the overall concept overview of light reaction, that is trapping the solar energy by the chlorophyll A pigment, and how electrons are getting excited in the chlorophyll A, and how the electron transport is taking place within the membrane, and how it is reaching the photosystem 2. And photosystem 1 is seen in non-cyclic photophosphorylation. Today, you are going to see how actually ATP is synthesized and NADPH is synthesized. Before that, I would like to appreciate the work of G. Pranita from Vangara TSRJC, Manasa and Bhuvaneshwari from Siddipeta. Very good, children. You have been following properly and writing the assignments in time and submitting very fast. Keep it up. I wish all other JLs also monitor the work properly and immediately send it to me so that I can mention their children name also. So let us get into the topic today. This picture is given for chemiosmotic hypothesis in your textbook. You are familiar with this, and anytime you can have a check. First of all, understand this picture. This picture is nothing but it is the thylakoid membrane. Thylakoid membrane or grana is nothing but stacks of thylakoid membrane. So such type of thylakoid membranes are located in the chloroplast. And in the chloroplast, you also see the colorless fluid, that is the stroma. Stroma is a site for dark reaction. And this thylakoid is a site for light reactions. So completely light dependent reactions are going to take place here in this thylakoid, and you have to see this one thylakoid membrane. It is having two membranes, outer membrane and the inner membrane. And the location of the photosystem 2, that is P680, and this is the location of photosystem 1, P700, where the chlorophyll molecules are going to form the light harvesting complex and harvest the solar energy, and electrons are going to get excited and they are going to jump from the valence shell, and when they jump, they are again coming down. The energy will be decreasing. So when they jump with more energy, high energy, and they'll be coming downhill, and they are getting passed from photosystem 2 to photosystem 1. And here you can see only cytochrome, but in the previous class I have mentioned about the plastoquinone and PQ, plastoquinone, and PC, plastocyanin. These two also exist here, but because of the extra work it does, it is mentioned here. So this PQ, cytochrome, plastocyanin are the electron transporters. They are only transporting, and it is called electron transportation, ETS, electron transport system. Sorry. So here, in this manner, the electron which is going to pass to the photosystem 1. After that, it is also having a reaction center at photosystem 1. Here also chlorophyll electron is going to jump, and ferritoxin is going to transport that one to NADP to make it reduce to NADPH. Sorry. So here, finally, the main concept of this is formation of this ATP, NADPH, and also photolysis of water takes place here in the inner membrane. Inner membrane of these thylakoids, the water harvesting complex, sorry, the water splitting complex, water is going to split and oxygen is going to get released. These three things are the important things taking place in the light reaction, and you have to learn about how ATP is synthesized, how NADPH is going to get synthesized. So for this, a clear vision of this thylakoid membrane, and the most important systems, the photosystem 2, photosystem 1, which is located and having the non-cyclic photophosphorylation taking place, and the important cytochrome is mentioned here. Apart from that, the new thing which you are noticing is a lumen. This lumen is the innermost part of this thylakoid membrane, and you also see here protons. Protons are also there, and the third thing which you see here is this. This is a protein molecule. This is a protein molecule having two parts. Protein molecule having two parts which is embedded inside. The part which is embedded inside is the F0 particle, and the part which is protruding outside is F1 particle. So you have F0, F1 particle. F1 particle is going to act as an enzyme, and what for this is located, you must know. You can see with the pictorial representation how the hydrogens are coming from stroma into the cytochrome. From cytochrome, it is entering into the lumen. Not only this, but also water splitting takes place, and those protons will also be present in the lumen. And how these protons are coming out? This coming out is making the photophosphorylation take place. This one. You can see how ATP is getting synthesized. So this thing you are going to learn. So clearly understand the structure. It will be very useful for knowing the concept here, how ATP synthesis will take place, how NADPH is going to get synthesized. Both these things, as you can see in the picture, they are located outside the thylakoid. That means they are present in the stroma. So light reactions getting taken place in the grana thylakoid membrane, but the formation of ATP and NADPH is in stroma part, because further the dark reaction, that means carbon dioxide assimilation, will be taking place in the stroma, and it requires energy. That energy it is going to get from the ATP as well as NADPH. Okay, so this is the thing. The connecting link between light reaction and dark reaction is NADPH and ATP. So let us learn how this thing is happening, how the synthesis is taking place in a clear way. The chemiosmotic hypothesis has been put forward to explain how actually ATP is synthesized in the chloroplast. So in the chloroplast, in light reaction, whatever energy is trapped by the sunlight uh, chlorophyll pigment molecule, that energy will be utilized for synthesizing ATP. ATP synthesis is linked to development of a proton gradient across a membrane, that is the thylakoid membrane. So simply this ATP synthesis is not very easy thing. For this, a proton gradient must be developed. A proton gradient is developed across a membrane, the thylakoid membrane. The proton gradient is going to develop, and the energy from the protons will be utilized for converting ADP to ATP. So this is the main thing happening, and the membrane role is very important. Thylakoid membrane role is very important. The proton accumulation is towards the inside of the membrane. That means in the lumen. So where are the protons present? These protons are present in the lumen of thylakoid, inside of the thylakoid. And when the gradient is high, from higher to lower concentration, the diffusion will be taking place. You know that diffusion is nothing but movement of molecule from higher concentration to lower concentration, or we can say higher gradient to lower gradient. So that lumen is the site where the proton accumulation, protons are more in number. The protons has to increase in number in the lumen, so that it will become higher gradient. From higher gradient, the protons will be moving to the lower gradient. When they are moving, at that time, the energy present in these protons will be utilized for the synthesis of ATP. So this is the thing happening, and this is a clear cut diagram of the same thing, the textual diagram, as just now you have studied. So once again, have a clear view of this diagram, how chemiosmotic hypothesis is taking place. You can see P680. Recall, what is P680? Photosystem 2. Here the electron, red color dot is representing the electron here. Its electron is becoming higher concentration, high energy level, and it is just moving to the P700. When it will move to the P700, it is moving through this membrane only. You can see from P680 to P700. When it is moving, you can see plastoquinone, cytochrome, and plastocyanin. These are present. These plastoquinone, cytochrome, plastocyanin are electron carriers. They are not using the energy in that. They are electron carriers. They are passing, only passing of electrons taking place, and electron is passed to the P700. Here also in P700 also the same thing is happening. The sun rays are falling on P700 reaction center. The chlorophyll A electron is also jumping, and when it is jumping, because of high energy state, ferridoxin, the electron acceptor is taking, and it is providing it to NADP. NADP. So ferridoxin is a electron transporter, electron transporter. Though we say these electron, they are accepting this one, they are not using for themselves, they are passing. When they accept primary electron acceptor, plastoquinone, you can see here, it is accepting and passing it to cytochrome. Cytochrome will accept that one and pass it to plastocyanin. Plastocyanin will pass it to P700, photosystem 1. Okay, so here the electron of P680 is going to P700. One thing happened here. Now P700's electron is given to FT, FT is giving it to NADP, the another thing. So electron is taken by NADP here. But what about the loss of electron by P680? This P680, it has given its electron. It wants its electron back. So what it does? It will break the water molecule. That is the photolysis of water. As you can see, the water molecule, water breaking, splitting of water molecule, that complex is located in the inner membrane. It is located on the inner membrane of thylakoid, and the oxygen and protons, electron is taken by this P680, and here the protons. So these three things are going to form when the water is splitting. Oxygen is a gas, so it will be diffusing out. But electron, this red color dot is nothing but the electron. It is taken by P680 because it has lost its electron. So it will be taking from the water molecule. So here the electron case is over. But what about this proton? This proton of water molecule is still there in the lumen. This light green color is the lumen. In the lumen, the water molecules protons are also there. Okay, so many water molecule protons are there. Not only these protons, but also the protons coming from outside. You can see here in this, it is given through PQ, but through the cytochrome it is going to enter here and enter the lumen. Okay, so the outside from the stroma also, when these plastoquinone, cytochrome, plastocyanin is passing the electrons to PS2 to PS1, at that moment they are utilizing some energy and taking a pair of electron from outside, that means from the stroma, and bring it inside of the lumen. So in the lumen, it is filling it. So the protons are getting filled from outside also. That means they are pumped. Proton pump. So these plastoquinone, cytochrome, plastocyanin, these electron acceptor, they are also acting as proton pump. They are pumping up the protons present in the stroma to inside of the thylakoid, that is in the lumen of thylakoid, where already the protons of water molecule is also there. So like this, the gradient of protons is increased here. The gradient of proton is increased here. So remember this thing, this proton gradient is increased. Now this proton gradient, when they increase, they want to move from here. They will move from higher gradient to lower gradient. Where is the lower gradient? The lower gradient is outside the thylakoid. That means again back in the stroma. So they want to move from lumen to outside. So when they want to move from lumen to outside, diffusion will take place. Diffusion does not require any energy. And the most important thing, diffusion will take place through the lipid molecules. But here we are having the protein because these water molecules, they want a protein carrier. The protein carrier is nothing but this F0, F1 particle. F0, F1 particle. F0 is embedded. Embedded means inside the membrane only. And F1 is outside. Protruding outside means it is outside somewhat. So F0, F1. So this is going to help in the facilitated diffusion. Facilitated diffusion means it is going to take these protons from inside the lumen and it is going to help it come out. And when it is passing through the F1, F1 is nothing but it is going to act as an enzyme called ATPase. ATPase. ATPase means ATP synthetase. Okay, so this ATPase or ATP synthetase enzyme is going to convert ADP plus inorganic phosphate to ATP. So here phosphorylation is taking place, conversion of adenosine diphosphate combining with inorganic phosphate and formation of adenosine triphosphate is taking place. So like this, the synthesis of ATP is taking place. This is one thing. And the another thing, when these protons are coming out, when they come out, what will happen? Here, you know that NADP, this NADP complex is on outer membrane of the thylakoid. It is not present in the inner membrane. It is present on the outer membrane of thylakoid. So this NADP already it received electron from ferridoxin. And after receiving that electron, it wants some protons also. The protons which are coming out of the lumen to the stroma, that protons will be taken here, and NADP plus these protons is going to get reduced to NADPH2. Okay, so this is the final end product of light reaction, NADPH2, ATP and oxygen. So these are the end products of light reaction, oxygen, NADPH2 and ATP are going to form. This is the overall concept about chemiosmotic hypothesis. And here this water molecule equation is also given here. Two molecules of water molecule, when it is splitting, it is given 4 protons, 1 oxygen molecule and 4 electrons. So this is what you are going to learn, how a proton gradient develops. As you know, for the proton gradient to develop, a water molecule has to split, one thing. And from the stroma also, a pair of protons are getting pumped inside the lumen. Two things are happening. So splitting of water molecule takes place on inner side of membrane, contributing protons of or hydrogen ions to accumulate in lumen of thylakoid. This is one thing. So proton gradient will develop inside the lumen because of splitting of water molecule. Is there any other mode? Means yes, other mode is also there. that is the primary electron transporter located towards outside of the membrane, transfers its electron to a hydrogen carrier, plastoquinone. Hence, it removes a proton from the stroma while transporting an electron. So, while transporting the electron plastoquinone to cytochrome, the plastoquinone is also passing the passing the protons, hydrogen, from the stroma to the inward lumen. Proton gradient increases. Now, what will happen? Proton gradient is increasing. Gradient means concentration is increasing due to quinone cycle, that is cyclic flow of electrons between plastoquinone and cytochrome B. Okay, so plastoquinone, cytochrome B, these are electron transporters as well as they are also transporting the protons from stroma to lumen. That means they are pumping the protons, hydrogens, ions from stroma to the lumen in the thylakoid. Now, NADP reductase enzyme is also important. It is located on the stroma side of the membrane. That means on the outer membrane of the thylakoid membrane. That means not inside the lumen, outside the thylakoid membrane. It is present, NADP reductase enzyme. So, there only the NADP is going to get converted or get reduced to NADPH2 by accepting electrons and protons. NADP will become reduced, and in which presence it is occurring in the presence of NADP reductase enzyme. This enzyme is also very important. Along with the electrons that come from the acceptor of electrons of PS1, protons are necessary for the reduction of NADP to NADPH2. How a proton gradient develops further, we are seeing NADP reductase enzyme is located on the stroma side of the membrane, and how from the PS1, photosystem 1, P700, electron is also jumping there, and that is taken by ferridoxin and given to NADP. Okay, and also the proteins, the protons which are in the stroma also increased, and both these are going to make the NADP get reduced to NADPH2, and that in the presence of NADP reductase enzyme. Within the chloroplast, the proton in the stroma decrease. So now what will happen? Within the chloroplast, the protons in the stroma decrease in number, while in the lumen, there is accumulation of protons. In the beginning, it happened. The protons from stroma is taken into the lumen. The water protons are also present in the lumen. That means they are accumulating more in number. So their concentration has increased. This creates a proton gradient. So the proton gradient formation is the most important thing taking place. And how the proton gradient is increasing means proton pump. Proton pump is nothing but from stroma, the protons are getting pumped into the lumen. That is also there. As well as measurable decrease in pH in the lumen. So the pH is going to decrease in the lumen. So this gradient is very important because it is the breakdown of this gradient that leads to release of energy. So when they accumulate more in number in the lumen, the protons, they must be removed. They must be sent out from lumen to stroma again through the thylakoid membrane. When they are sent out, at that time only the movement will take place and their energy will be lost. The energy will be reduced. The energy will be coming out. That energy will be utilized for ATP synthesis. So here one important topic, facilitated diffusion. In the first chapter, which is deleted one for you, in that you will be having this concept of facilitated diffusion. Already previously I asked you to revise the lesson, first chapter and second chapter, so that you will have an idea. The diffusion of any substance across a membrane depends upon its solubility in lipids, which also move faster. So for any substance to diffuse from one medium to another medium, it has to pass through certain membranes. in biological systems. And when it has to move from a membrane, it must get soluble in the lipids. But hydrogen protons, these are not going to move. They require protein carriers. These are not going to get soluble in the lipids, so they won't move through the lipids. They require proteins. So substances which have a hydrophilic moiety find it difficult to move through the membrane. Their movement has to be facilitated by membrane proteins. Membrane protein, that is F0, F1 particle, as you have seen already, without utilizing metabolic energy, and there must be a concentration gradient, means from higher concentration to lower concentration it has to move. And this is only called as facilitated diffusion that is taking place. So this one is a particle. This is a protein particle F0, F1. And this F1 is going to act as an enzyme. ATP synthase. So this F1 particle, which is going to act as an enzyme, when these protons which are present in the lumen is passing through F0, F1, at that time certain energy is going to get liberated, given off. From higher gradient it is moving to the lower gradient. Lower gradient is present in the stroma now. Higher gradient is present in the lumen. So when these protons are moving, they are losing that energy, and that energy could be utilized for converting ADP to ATP. So in this manner, it is taking place. You can see clearly here. This is the thylakoid lumen where proton accumulation or higher gradient is there. And this is the stroma of the chloroplast where protons are less now. So there is a gradient difference. From higher concentration, it has to move on to the lower concentration. So it cannot move through the lipids. So it will take help of these protein molecules embedded. These protein molecules are the F0, F1 particle. F0 is embedded inside the membrane and F1 is protruding outside. So through this F0, F1 particle, the protons are coming out. Meanwhile, the energy which is given off when they are coming will be taken by the ADP and inorganic phosphate, and they are going to get converted into ATP. Okay. And the enzyme for this conversion is ATP synthetase. So in this chapter, in this topic, we have learned about ATP and NADPH synthesis. Let us see once again. F0 helps in facilitated diffusion. That is the most important requirement. It is this F0 which is helping in facilitated diffusion. The protons are coming out from lumen to stroma. And here only the ATP synthesis is taking place. ATP is getting formed in the stroma. Remember one most important thing, the ATP is getting synthesized in the stroma. NADPH is also getting synthesized in the stroma. Okay. Photolysis complex is located on inner side of thylakoid, and photolysis complex means water is splitting that is occurring in the lumen inside the thylakoid membrane on the inner side, and there only the proton gradient can increase because of this photolysis of water. And next, NADPH2 complex is also located on outer membrane of thylakoid. This is the thylakoid, this is the lumen. Water splitting complex is inside, NADPH complex is outside. Okay, and F0 particle is going to help in facilitated diffusion. Okay, so here ATP is forming in the stroma only, NADPH2 is also forming in the stroma only. Okay, so these two things you must remember. And photolysis is inside the lumen, so oxygen is also forming in the lumen only, but oxygen is a gas, it will be diffusing out, coming out. So these are the important things taking place in the light reaction. So oxygen is released in the lumen, ATP and NADPH are synthesized in the stroma, proton gradient is high in lumen, ATP synthetase or ATPase enzyme catalyzes the formation of ATP. Chemiosmotic hypothesis is nothing but it requires a membrane. And what is that membrane? Thylakoid membrane. A proton pump. A proton pump is nothing but PS1 to PS2 to PS1 when the electrons are moving through the electron transport carriers. They are only pumping the protons from stroma to lumen. So a proton pump is developed by the electron transport molecules only. The electron transport molecules are plastoquinone, cytochrome, plastocyanin. So they are only when they are passing the electron from PS2 to PS1 from the stroma inside the lumen in the thylakoid membrane, they are pumping some protons. So here a membrane is a thylakoid membrane, a proton pump is also there, and a proton gradient is increasing because of photolysis of water also, and F0 F1 particle is also very important for the ATP synthesis. So that's why this simple and easy picture is given in your textbook where you can see the PS2, that is 680 nanometer wavelength, PS1 700 nanometer wavelength, and you are seeing how the electron from PS2 is moving to PS1, that is through the electron transporters here. The role of this electron transporter is not just transporting the electrons, they are not just the electron acceptor molecules, they are also pumping the hydrogen from stroma to the lumen. You can see the green arrow indicates how the protons from stroma is pumped into the lumen. Okay, as well as there will be water complex splitting of water molecules, that is also adding the protons here. Okay, and when these proton gradient is increased here, that protons are coming out, and F0 F1 particle is also having their role for the synthesis of ATP, and the synthesis of ATP is taking place in the stroma. And when these protons are coming out, what happens to these protons? They are in the stroma. They are in the stroma means they are going to combine with the NADP, which already took some electron from this photosystem 1, and the protons are added and it is getting reduced NADPH2. So here NADPH2, ATP are the end products of light reaction, as well as oxygen which is formed in the lumen. Oxygen is formed in the lumen. ATP and NADPH are formed in the stroma, which further helps in the carbon assimilation or carbon fixation is going to take place in the stroma. Simply we will be saying dark reaction, but it is better to call it as biosynthetic reaction. Today's assignment is mention the components of ATPase enzyme. What is their location? Which part of the enzyme shows conformational change? Where does the photolysis of water occur? What is its significance? Only two answers. I am not giving heavy work, heavy assignment. Simple, very easy and very, very important as per IPE. Those questions are given as assignment. It is better you write and memorize it properly and forward your work to your JLs so that they'll forward it to me, and that will be a motivation for you when I am taking a name. Revise the textbook, and you can also revise the uh, topic by watching the videos again and again, because these are uploaded videos. You can have a clear cut understanding. Sometimes certain topics on the first time only you can't understand. If you understand also, you cannot remember or you cannot recall. So frequently watching these videos or else revising the textbook is going to help you for better understanding and better recalling of the topic. Okay, thank you students. Thank you sir. End of the session.